The cell. Dun, dun, dun. So, a cell is the smallest unit of life, the smallest unit capable of performing the functions of life. Within the cell are membrane-bound structures. So these are little things in there that have membrane around them. And this is part of that compartmentalization of life. And these little structures are referred to as organelles. And there's different organelles and such. And the ones that you can actually see with the microscope, that would be the nucleus. The nucleus is unique because it contains the DNA. It also is two membranes. It's double membrane. You have another organelle, the mitochondria, which is double membrane. And it also contains DNA. It contains its own mitochondrial DNA. But there's another structure within the nucleus we'll see here in just a minute called a nucleolus. And it is an area where ribosomes are made. Ribosomes are produced from RNA, which is a nucleic acid. The other nucleic acids are formed within the nucleus in a process known as transcription. So messenger RNA and transfer RNA and ribosomal RNA are all produced within the nucleus in a process known as transcription, and they're all needed for protein synthesis and all. So the DNA itself, it is a double-stranded molecule. They complement each other. There's 4 nucleotides there, and we'll go into that in another little quick video that I have for you. And you have the notes, I believe. So, but that nucleolus, nucleoli is found in there, and it's the source of ribosomal RNA. So where are ribosomes produced? They are produced in the nucleolus. Ribosomes don't have a membrane. This is the one organelle that is similar in both eukaryotic, which is you and I, and prokaryotic, which is bacteria. So we have ribosomes and bacteria have ribosomes. Your mitochondria has its own ribosomes. They're different in size between the species, between the eukaryotes like you and I, trees, fungi, and bacteria. But bacterial ribosomes are similar in size to mitochondrial ribosomes. And when you get into microbiology, and we've got to get into how antibiotics work, some antibiotics actually have a negative effect on your mitochondria. So, but the ribosomes, they're produced in that nucleoli. They leave the nucleus through these openings called nuclear pores. They go out into the cytoplasm where they can be free, or they go and attach to this membrane, this large internal membrane known as the endoplasmic reticulum. And when they attach to that, that ER is referred to as rough ER. It's at the ribosome where proteins are synthesized. So the ribosome is the site of protein synthesis. Go back to that ER. There's 2 flavors. There's a rough ER and a smooth ER. The rough ER is closer to the nucleus. The smooth ER is closer to the membrane. The rough ER is where the ribosomes are attached and proteins are produced, and then the proteins which are produced travel through this internal highway toward a structure known as the Golgi apparatus. The smooth ER, which since it's closer to the membrane, as materials are brought into the cell, it can then detoxify these substances, and it can even start the process of glucose metabolism, this called glycolysis and such, and then also lipids and such are produced or synthesized in the smooth ER. I mentioned that Golgi body. An older term for it is a dictyosome, but the Golgi body, it's a series of membranes. They're all stacked up. They're flattened stacks of membranes and such, and these proteins enter in on one side, and the proteins are processed. They are folded, they are shaped, they are rearranged, and then they are wrapped up in membrane and shipped out. When they wrap them up in these little membranes, these structures, those are vesicles and such, and then they are secreted. Now, depending upon what's inside the vesicle, you have different types called lysosomes and peroxisomes and such. But that's how these proteins are produced and shipped out of the cell. Some things are brought in, and then some things are shipped out. And I told you before, when the cell's drinking, that's penocytosis. When it's eating, that's phagocytosis. Things come in, things come out, endocytosis, exocytosis. One type of protein that's produced are digestive enzymes, and these are very dangerous enzymes because they will digest anything. So the cell wraps them up in a membrane, and that protects the cell. If that lysosome was to rupture, it would auto-digest. It would self-digest the cell. And that would cause cell death, apoptosis. Sometimes when you freeze food, the water within the cell forms crystals, and these crystals grow, and they can puncture these lysosomes. The enzymes leak out and start to digest those cells. That's what causes freezer burn. A very important organelle within the cell is the mitochondria. It's the site of a majority of ATP synthesis. Not all ATP is produced in the mitochondria, but a majority of it is when oxygen is present. When oxygen is not present, the mitochondria can't function. They contain their own enzymes. They contain special proteins, these cytochromes. It's where the Krebs cycle takes place. It's where the electron transport chain is located. You get your mitochondria from your mother. It's called maternal mitochondrial DNA, your maternal mitochondrial DNA. She got it from her mother. She got it from her mother, and on and on and on and on. You can read about this. You can research this, and it's referred to as a mitochondrial Eve. And it takes us back some 200,000+ years into central or the equatorial regions of Africa. When you exercise, when you demand more ATP, your body will make more mitochondria. You don't have to make more cells because these mitochondria are kind of like bacteria, and they will just make more. Now, when you make more mitochondria, you have more powerhouses. You have to put more fuel in, and there you have to keep putting fuel in there to get more energy. The theory is if you do this, your body will then keep shoveling fuel into that, and if you're not ingesting that fuel, your body will start going to your fat stores and converting the fat back into glucose, and then feeding these mitochondria. When you ingest a lot of calories and you don't have enough mitochondria, or when all of the Krebs cycles are full, your body takes those molecules that you ingested, and then it runs through a process and it puts it back into your adipose tissue or your fat stores. Other modifications that a cell might have are cilia or flagella. There's only one cell in humans that have a flagella, and that is the sperm, and that's for motility. Other cells in both male and female, they are ciliated, and these cilia are on the apical surface, the top surface of the cell, and they are used to move particles. Looking at a cilia and a flagella, if you cross-section them and look at the little bitty fibers inside of them, you would note there are those microtubules. There's different arrangements inside there. That's how you know one's a cilia and one's a flagella. But it's for movement. An analogy that I was taught a long time ago, think of the flagella as just you in a canoe with one paddle. Whereas cilia were a lot of people in a big canoe with lots of paddles. It's just how it's a smoother movement and all. But in your throat, in your trachea and such, you have ciliated cells, and in the fallopian tubes of the female, they have ciliated cells. Other structures are these centrioles. Animal cells have centrioles. Plant cells don't. These centrioles are just an area where these internal tubes will be formed and attached, and it's needed during cell division, mitosis and meiosis. So these are made of the same microtubules that the cilia and the flagella are made of, but it's just there. All cells have this. Another structure of microvilli. Microvilli are little bitty fingers that increase the cell surface area. So where you need absorption, like in your digestive system, you have microvilli. There's other types of microvilli, stercovilli and such there. So you can read about those. The internal framework of the cell, you have these microfilaments. So it's like a scaffold that holds the cell up and anchors the organelles into place. And so cells have that. They have this internal skeletal system. Sometimes these fibers pass between cells and it kind of holds the cells together. And these demisomes and hemidemisomes and such, they create these kind of like rivets that hold cells together. It creates these junctions, and then you have different types of junctions between different types of cells and tissues in the body, where if you don't want things passing through easily, you have really tight junctions, and if it's okay for stuff to pass through, you have like it's not as tight, or you have gaps in the junctions and such there. So I mentioned that tight junction to you. It just depends on what do you want to be allowed to cross that barrier, like in your kidneys or in your lungs or in your digestive tract or in the brains, meninges and such. So a desmosome is a type of anchoring junction, and it kind of anchors two cells together. So they call it a macula adherens. It's a sticky spot. So macula means spot. So the gap junctions. In some special tissues, like in your heart cells, you have these open areas. It's kind of like a porthole between the cells, and that allows these electrical signals that are generated on one end of the muscle, the heart, to travel across and jump across these. So really, all of the cells kind of respond at the same time and such. So a good example of them are found within the cardiac muscles. Smooth muscles have them as well. Your smooth muscles kind of react in the area as one unit, such, but the cardiac's a better example. There we go. We went through the cell.